I hope that you guys are having a good start to your day. I hope that your week's going pretty well as well. Um, we are going to go ahead and jump off from where we were just the other day when we were talking about carbonic anhydrase. Um, remember, we were talking about basically the movement of oxygen throughout your body, using hemoglobin and myoglobin as models for protein ligand interactions. Um, so we're going to continue that. Share my screen right about now. And then whenever I'm wrapping up lecture, I will share something else with you, kind of what to expect over the next few days related to some of the things I've been working on. Okay, so what we have is basically just another model that I like this model because it, I think, provides a little bit more insight and detail about what's happening. So we're moving oxygen from the lungs into the muscles, and then we're moving CO2 out because we're generating CO2 when we're breaking down when we're completing the breakdown of sugars and breaking down fats. Um, I always think of fats just because there's a process that we're going to learn about in a couple of days called beta oxidation. And beta oxidation, which is where you generate a huge amount of CO2, and a huge amount of energy from a fatty acid. Um, so just to kind of follow this process, we're inhaling oxygen, and that oxygen in our bloodstream is going to hit a more or less deoxygenated, or for our purposes, we'll refer to it as a deoxygenated hemoglobin, making our oxygenated hemoglobin. That is going to have a total of four O2s on it. Our pressure of O2 in the lungs is approximately 100 torr, so it's super high. So even subunits of hemoglobin that have a low affinity for O2 are going to be able to bind it. So that HbO2 is going to go through our bloodstream, and ultimately it's going to arrive at our muscles where that O2 is going to be deposited and released. And it's going to be released because of the partial pressure of O2 is approximately 20 torr. These are rounded numbers, but effectively what that means is that our oxygen concentration is relatively low compared to in the lungs. And so hemoglobin is going to say, oh, okay, well, that the oxygen concentration in the tissues, in the muscles, is considerably lower than my P50. So what I'm going to be able to do is release that. And so we're going to drop that O2 off. The fate of that hemoglobin, well, it's now kind of deoxygenated. It's back in the bloodstream and it's just kind of hanging out. While in the muscle, what's taking place is this O2 can be picked up by myoglobin, MB, and make, or a single myoglobin protein, make an oxygenated myoglobin, and basically hold on to that O2 until it needs to release it. That O2 is then going to go up through, or not up necessarily, but it's going to be imported into the mitochondria so it can participate in respiration. And if you've ever taken like a cell biology class, or maybe even like a biology class dating back to like high school or something like that, you probably learned about the Krebs cycle. And if you didn't learn about it at the Krebs cycle, maybe you learned about it as the TCA cycle. The TCA cycle standing for tricarboxylic acid cycle. And then maybe even beyond there, the final part where you're actually using that oxygen, you probably learned about something known as oxidative phosphorylation. Now, it's in those processes and pathways where we're actually going to be utilizing that O2 and generating that CO2, or generating CO2. And whenever I say generating that CO2, I'm referencing, of course, this CO2. CO2 is generated in the muscle, and an enzyme known as carbonic anhydrase in the bloodstream is going to take that CO2 and react it with H2O, and it's going to produce HCO3 minus and a singular proton. Now, that proton, well, as is the case whenever we're thinking about an amino acid and a proton, depending on the pKa. and depending on the concentration of those protons, the question is, well, is that proton going to go on to one of the functional groups of an amino acid? And we know that histidine 146 is an amino acid that was implicated as being particularly important for what's known as the Bohr effect. So that's going to be capable of binding onto histidine 146, which is going to favor the T state of our deoxygenated hemoglobin. This HCO3 that's generated also from the carbonic anhydrase reaction can bind to the N termini of, I'm going to say HB, and I'll say N term, not firm, N termini, N termini of hemoglobin. It's going to jump on there in what's known as a carbamation event. So we have this protonated hemoglobin that's going to move through the bloodstream. We have our carbamated hemoglobin that's also going to move through the bloodstream. And ultimately, both of these things are going to arrive close to or in the alveoli where they can react, and carbonic anhydrase can take those two things and produce CO2 and H2O. That CO2 is then going to be exhaled, which is also going to give us back our hemoglobin that can then bind O2 again in that oxygen-rich environment of the lungs. So what I think is helpful to think about throughout all of this process is to think about the different states, to think about how dynamic hemoglobin is, and think about things like the P50 or hemoglobin's P50. Hemoglobin's P50 is approximately, if I remember correctly, it's about 26. That's its overall P50. So when you are, if you gave hemoglobin a oxygen concentration or a partial pressure of oxygen of approximately 26, well, then 50% of the binding sites would be occupied. Now, that's whenever I apologize for any sort of gray area of this terminology, but sometimes I'll refer to a singular hemoglobin protein because it's easier to understand kind of what's happening there molecularly. But then that's not really realistic for what's taking place in your body, and what's taking place in your bloodstream, because in your bloodstream, you've got a concentration of hemoglobin proteins. You have a large number of hemoglobin proteins. So whenever I say 50% saturated, what that means is that 50% of the binding sites in a mixture of hemoglobin proteins. So I'm going to come over here. If we're talking about 50% saturated, I'm going to say hemoglobin population. Well, that population, each member of that population has 4 binding sites. So whenever we say 50% saturated, that can mean a number of different things. That's not saying that every single hemoglobin protein has 2 oxygens bound, but instead the population is 50% saturated. So you might have a mixture of HB, O2, 4. So one hemoglobin protein that has 4 O2s bound, and then I'll just put in parentheses 3, parentheses 2, parentheses 1, and then, well, effectively 0, where you have just the free HB. I just want to bring that up as a point where you have to kind of jump between the molecular level and the kind of more macro level. So oxygenation has an effect on releasing a proton. Releasing a proton is going to shift the equilibrium from bicarbonate to CO2 and H2O. And once we get back here, that's when our CO2 can actually be released and expired from our body. Oh, we got our first question, and this question is duplicated as an assigned question. I believe it goes up at 9:50. And remember, I'm looking for everyone to. I had an emailed question yesterday about Top Hat, and I haven't added last week's questions. I'm trying to get those going. But if you do not do the questions in class, that's fine. I'm looking for everyone to do at least one. I want responses so I can gauge our understanding of it. Um, but then if you elect to not participate in them, I need you to do at least one for the assigned questions. The assigned questions are up until tomorrow at 11 a.m. Okay, so CO2 is added to the N-termini of hemoglobin, is best known as a, you got about 60 seconds. It's registering. There we go. 57 seconds. 15 seconds left. All right, let's see where we are. We had 50 people in class and 50 people answered. Great. So where are we? A carbamation. Okay, carboxylation is a super tempting term, but this is kind of a unique, you could look at this as it's a unique carboxylation. So that's why the best term for this is a carbamation. Carbiding was just kind of a decoy answer out there. And that is a carboxylation that goes to the N-termini of the protein. So I was looking for the best answer there, and so the best answer is carbamation. Now, what happens in the tissue? This is kind of bringing a couple of different things into play with respect to the Bohr effect. Carbon dioxide is abundant and the pH is low in the tissues. And so as a result of that high CO2, we're going to have a lowered pH that's favored, and that's going to favor the release of O2. I think that it's helpful to, when you see something like this, to put it in the context of the T and the R state. So remember, our R state is our um, active form of our protein. Sometimes the terminology that I heard as a graduate student for T and R is that the T state is kind of referenced as the tense or the yeah, T for tense or unreactive state, whereas R is more relaxed. It can bind onto its substrate or ligand and release its ligand, and it does so a little bit more freely. So the formation of HCO3 minus releases more protons. Reaction with hemoglobin releases more protons. And favoring the deoxy or the T state uh, releases O2 and binds CO2. So this graph, what we're looking at here is you could think about it as our normal conditions as the line that's in the middle. One thing that I mentioned last time and I want to bring back is the shapes of these curves, though they might look subtly different, think about these curves as effectively all being the same. They're all that sigmoidal shape as opposed to a hyperbolic shape. Now, these curves display a shift to the left or to the right. If we're shifting from the normal conditions to the left, the points that are going to take that to the left are a decreased hydrogen ion concentration, which is an increased pH, a decreased CO2 concentration, and a decrease in temperature. Now, we hadn't talked about temperature, but you could think about temperature effects as something that you're going to study in any sort of enzyme. Any enzyme that you ever want to characterize, you always want to look at, well, how does the temperature impact this enzyme's activity? Now, a shift to the right is highlighted primarily by, and we're already familiar with these, as what's in the tissue, where you have an increased hydrogen ion concentration or a decrease in pH and an increased CO2 concentration. Now, I want you to be able to comfortably code switch between hydrogen ion concentration and pH. I know that that's something that you definitely saw in Gen Chem 2, but it's an important skill that it's like a subtle detail, and it's almost like a trick question where you read the question, it says an increase in pH means blah. Okay, well, an increase in pH, that means it's more basic. And so I personally have a tendency to go in that direction. Okay, got it, more basic. And if I'm reading question after question or statement after statement that talk about a change in pH, then I might get kind of a one-track mind. And then if you throw a question at me or a statement at me that addresses hydrogen ion concentration, I might have been so ingrained in this conversation or thought about pH that I might kind of run into a little bit of a wall and be like, oh yeah, I know an increase in hydrogen ion concentration is a more basic environment. And that's the exact opposite. So I want you to feel comfortable, and I'll go ahead and rewrite this, kind of code switching between hydrogen ion concentration and pH. Make sure that you're comfortable doing that. Low pH means acidic. A low pH means high hydrogen ion concentration. Okay, so after this, we have this new compound, bisphosphoglycerate. Now, bisphosphoglycerate is a naturally occurring compound. This is known as BPG or D23 bisphosphoglycerate. The capital D, that should be a little bit familiar because we talked about um, L and D sugars, and bisphosphoglycerate is an example of the D sugar. Now, the L sugar, I think that, I don't know. I've not looked this up, but I would imagine that, well, one, I know that L sugars are not naturally occurring. They're very, 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 very rare across every microorganism and every organism that's known. They can be synthesized, but L23-bisphosphoglycerate, I would bet does not have the same effect as D23-bisphosphoglycerate, because that's pretty much a trend. Whenever you compare a D sugar and an L sugar, the L sugars are not as, relevant biologically. Okay, so this is a naturally occurring compound in red blood cells. Now, one of the things when we first started talking about hemoglobin was we talked about that central cavity within the T state and the R state. Now, add to the chat, does anyone remember, was it the T state or the R state where that central cavity was larger? One of them, we got T state. You're exactly right. It is the T state. So the T state of hemoglobin had that like larger cavity. And effectively, what bisphosphoglycerate or 2,3-bisphosphoglycerate does is it enters into that central cavity and more or less locks it up. Now, its purpose is to modulate hemoglobin's oxygen affinity by binding preferentially to that deoxy or that T form of our protein. Now, 2,3-bisphosphoglycerate, if you're hearing that term, 2,3-bisphospho, I mean, my first thought is, okay, it's probably got, well, two phosphate groups. It binds inside the central cavity of deoxyhemoglobin, or our T state. 2,3-bisphosphoglycerate has a charge of -5. Okay, so this is our T state. It has a charge of -5. And so here what we're seeing is bisphosphoglycerate, don't worry about it, Milo. It interacts with that central cavity and with amino acids within that central cavity. Now, those negative charges on our 2,3-bisphosphoglycerate interact with a pair of lysines, 4 histidines, and 2 N-termini. And you might be looking at that and thinking, okay, 2 lysines, okay, 2 positive charges. 4 histidines, okay, 4 roughly positive charges. 2 N-termini, 2 positive charges. That would round up to be approximately 8 positive charges. So how do 5 negatives coordinate with 8 positives? Well, I think one thing that's worth thinking about is that those histidine residues, if this is at a physiological pH or a neutral pH, those histidines, you might have at any given point in time, you might have 3 that are positively charged and 1 that's neutral, not negatively charged, neutral. So that's one thing to consider, that basically these groups are going to kind of adopt the charges that they need to in order to interact with this bisphosphoglycerate. So bisphosphoglycerate, we've got a nice picture of it down here with those negative charges. Those negative charges are on our carboxylic acid, hence the glycerate, and we got two on each phosphate and one on our carboxylic acid there, because this is going to be an oxidized or reduced sugar. Um, Now, what I want you to know about bisphosphoglycerate is I want you to know that it brings in negative charges and it interacts with positive charges in the central cavity of the T state. Now, what that means is for the R state, I think that it's helpful to remember that the R state is something that's like it exists, but it's a little bit more fluid in the sense that that R state is not necessarily set in stone. And personally, the way that I think about bisphosphoglycerate and the R state versus the T state is I think about bisphosphoglycerate as being a molecule that's like it's kind of I don't know a better term for this, but it's chomping at the bit. It's ready to enter that central cavity of the R state of the protein. And as soon as it sees an opening, and whenever I'm thinking of, okay, let's say that we have 100 proteins and our saturation is approximately, I'm just going to say 99%. It's very, very high. Our O2 saturation is very, very high. Now, one thing that we've talked about, or one thing that I've brought up, is that this is never permanent. That 100% saturation is kind of never really attained. So I always kind of think about it as we've got 100 copies of hemoglobin, and there's 99 of them that have four O2s bound, and there's one of them that has three O2s bound. Well, that protein, or maybe it has two O2s bound, that one protein, it kind of leaves a little bit of an opening so that bisphosphoglycerate can kind of insert itself. And as soon as bisphosphoglycerate gets in there, then it's almost like it kind of makes room for itself and causes that protein to shift more towards the T state of our protein. So it's a one bisphosphoglycerate molecule, and I know that I'm talking about 100 copies of the protein and one copy of this molecule, but as the concentration of bisphosphoglycerate goes up, what's that going to do? That's going to shift our equilibrium from favoring the R state of our protein to the T state of our protein, because all of this is all about equilibrium. Whenever I said we had 100 copies of the protein, one of them was able to take on bisphosphoglycerate. Now that form of the protein, or that protein, is arguably in the T state. So what is that remaining population going to do? Well, they're going to try to maintain their equilibrium. And so one of those proteins is going to drop two or maybe one O2 molecule. And so then we're still going to have that partially um, deoxygenated protein. If there's another bisphosphoglycerate, it inserts itself, shifts it to the T state. So that's the way that is helpful for me to think about what's taking place here. It's almost like bisphosphoglycerate, as the concentration of it creeps up, it picks off one by one by one by one of these hemoglobin proteins and picks off the R state protein and transitions it to the T state protein. So I like this graph right here because I think that it brings up a number of different things. Um, if you were to have a completely stripped hemoglobin, so a hemoglobin protein devoid of an acidic pH, this is at a neutral pH, you would see something that looks like this right here. This would be your stripped hemoglobin. Your P50 would be approximately, here's my 50% point. I'm going to jot that down to, I'm going to call that roughly 9 So our P50 for our stripped hemoglobin is about 9 torr. Okay, so this is all being done in vitro in an in vitro setting. Now, this is like a really great experiment because effectively what we're doing is we're adding back other components. So the first thing is let's bubble a little bit of CO2 in there. And what does that do to our hemoglobin? Well, keeping in mind, this is not looking at a entire system. Instead, and I bring that up because does this line right here, so I'm going to underline or put a box around HB plus CO2. So this is purified hemoglobin, carbon dioxide, and oxygen. There's no carbonic anhydrase here. So what that means is that CO2, we're not seeing the drop in pH there, or the dramatic drop in pH that's caused by um, the carbonic anhydrase reaction. Instead, what we are seeing is the carbamation. So CO2 in the presence of O2 and hemoglobin is going to cause the P50 to go up. So simply by the addition of CO2, when you're holding O2 concentrations constant, you're going to see that P50 go up to approximately, I'm going to call that 18. And these numbers aren't of huge significance for precision, or the precision of them is not of huge significance. So we've got our stripped hemoglobin with a P50 of 9. Then, whenever we have, or when we compare that to, sorry, I'm toggling between my tools here, hemoglobin and just bisphosphoglycerate. Well, then our P50 is going to go up. I'm going to just call that 20. So if you're to think about or compare what has a more dramatic effect on hemoglobin's binding affinity for O2, you could say BPG has a greater effect, is greater than CO2. Now, the last line that I want to highlight is this black line, which corresponds to the hemoglobin plus BPG plus CO2. So there effectively, we have a system where we're trying to reconstruct everything that makes up everything that's in the bloodstream. Here, what we have is an increase where basically hemoglobin is, or the effects of bisphosphoglycerate and CO2 are additive on hemoglobin's oxygen binding affinity. Now, after it's all said and done, when we compare that to uh, whole blood, which is represented by this red line right here. Now, my first glance at this is I look at the two lines, the red line and the black line corresponding to hemoglobin, bisphosphoglycerate, and CO2. And I say, well, they're kind of right next to each other. But they're not right next to each other. And so there's like the skeptical eye in my head that says, well, they're not the same. They look similar, but they're not the same. So that to me means that there's some other factor in the whole blood that is in addition to bisphosphoglycerate and CO2. Maybe that's the effect of carbonic anhydrase. Maybe. Um, but then there's another part of me that's like, well, they're very similar to one another. I guess a little bit of the less critical or skeptical eye, and I'm like, oh, they're very similar. And I would say that both of those interpretations, not that everything, not to be all Pollyanna that everything's great, but to consider both of those interpretations, I think is valid. And the reason is because we don't see error bars on this graph in any form or fashion. And so it could be that this is within a margin of error there, that the difference is merely a simple error margin. I have not looked up this paper, the one that's highlighted down here. That paper right there from physiology. I've not looked up that paper, and maybe I should do that someday. But at any rate, what I want you to take away from this is one, that bisphosphoglycerate has a greater impact on hemoglobin's oxygen binding affinity than CO2, but both of them have a pretty stark impact on oxygen binding, or the oxygen binding affinity of hemoglobin. Now, this is like I I kind of previewed this a little bit. And I previewed it in the sense that I talked about your oxygenated hemoglobin and your deoxygenated hemoglobin. And well, that's not really the case. And this graph is a little bit complicated, but this, I think, highlights an important detail about bisphosphoglycerate and oxygen binding of hemoglobin. Now, what I'd like to draw your attention to first is the singular black line, the black curve representing our hemoglobin's oxygen binding affinity at sea level. Now, what we see here is the difference between um, our arterial CO2 level and our venous CO2 level. Okay. Now, our venous CO2 level, or sorry, our venous O2 level is it's approximately 30 torr. Okay, 30 torr is a little bit above our overall P50 for hemoglobin. And essentially what that's saying is that the difference between our arterial um, O2 level, arterial O2 level at sea level, or yeah, versus the venous PO2, what we see is approximately a drop of 0.38. Another way to interpret that is 38%. So basically, your oxygenated blood versus your deoxygenated blood. So let me get my pen. Okay. So your oxygenated blood, whenever the O2 concentration in the environment is 100, you're basically as saturated as you're going to get. Measuring the O2 concentration of the blood that's coming back from the tissues, the blood that's in your veins, well, it's about 60% saturated, really about 62%. What this difference means, so I'm referencing this point right here and this point right here, is hemoglobin, kind of as a population, drops off about 38% of the O2 that it picks up. So whenever I say that the deoxygenated form of hemoglobin, this is what I'm referencing. It's really 62% saturated. So it's not completely deoxygenated, it's partially deoxygenated. Now, again, bringing back the idea of what that looks like. You might have some proteins that lack all four oxygens. You might have some that remain completely oxygenated. But I kind of think about it as like a dump truck. 38% of its what's the word that I'm looking for? It starts with a C. Um, no. 38% of its materials and 38% of its cargo. That's the word that I was looking for. I was thinking carboy, but cargo. 38% of its cargo gets dropped off. Okay. So this is what hemoglobin does whenever you go from 100% or you're in an environment where the oxygen concentration is very high. Now, what I want to do is erase some stuff. I can erase our oxygenated. Now, if you decide to travel to the Mile High City, or if you want to go to Rocky Mountain National Park or something like that, well, chances are that you're going to go to a place that has an elevated altitude of approximately 4,500 meters. So that would be about, well, that would be higher than 4,500 meters would be, we're looking at close to 2.5 miles up. So certainly not Denver, because Denver's the mile high city. So let's say that we've gone and we're climbing Mount Rainier. There we go, keeping it domestic in 48 states. So you're climbing Mount Rainier. Well, when you get up to 4,500 meters, your altitude's at 4,500 meters, the O2 concentration in the environment is only about 50 torr. So the O2 concentration is about 50 torr. And what that means is that every time you inhale, you're giving all of the red blood cells in your lungs less oxygen than they need. And so you are starting right here. What that means is that your best O2 saturation is approximately, let's see, I'm going to call that 92%. This is about 92%. Now, if you've ever been to the emergency room or a doctor's office and gotten one of those pulse oximeters on your finger, or if you have a small child, they put them on the child's palm or on their foot. You're going to measure at about 92%. Your doctor is going to be like, oh, that's not good, because a good number is about 95% or above. 92% momentarily is not horrible. Not a physician, but that's just from my observations. 92%, that's not particularly good. And so effectively, you're starting out when you're at 4,500 feet or meters, 4,500 meters above sea level, you're starting out not a particularly good state. And so every time you inhale and exhale, you're not taking on enough oxygen. Now, what that means is, well, in kind of a vacuum, and not an actual vacuum here, but what you're going to do is you're going to take on less oxygen, and then, well, your tissues, hemoglobin's going to behave the same way. And it's going to get to the tissues and it's going to be like, listen, I've got less oxygen. This is what I can deposit. This is what I'm going to drop off. So when we follow that black line in a bisphosphate a BPG devoid environment, that's what hemoglobin's going to do. It's going to be able to take on less oxygen, and it's ultimately going to be dropping off less oxygen because it took on less. So altitude, HB, drops off less O2 because B/C, it picked up less. Okay. Now, what I'd like you to look at now is that red line. Okay, our red line is a little bit different. What our red line reflects is whenever your body says, oh, snap, the O2 concentration in our environment is low. So you know what we're going to do? We're going to make that naturally occurring compound bisphosphoglycerate. So I'm going to up the BPG concentration. And what is that going to do? Well, that's going to favor the T state of our protein. What that's also going to do is it's going to make it so that at that same oxygen concentration, that 50 torr, hemoglobin's going to be able to pick up less oxygen. Whenever it picks up less oxygen, you'd think, oh, that's a bad thing. And it is. But bisphosphoglycerate helps out, because what bisphosphoglycerate enables hemoglobin to do is when it picks up less oxygen, it's able to drop off more oxygen. So effectively, bisphosphoglycerate in a high concentration is a good thing because of the fact that it enables hemoglobin to drop off more oxygen at the tissues when there's less oxygen available to it. It's going to do that, and that's not particularly good. A perfect scenario is where you have a very high oxygen concentration, but in the event that the environmental concentration of oxygen is low, your body compensates and makes more bisphosphoglycerate. Now, I think a good one thing I don't know about this is how quickly this turns on. Um, I would imagine that it's probably pretty rapid, but I would also imagine that that rate is probably within like, a handful of hours. So you might have, and maybe this is implicate or this can be related to like altitude sickness. I don't know if anyone ever gets that. I one time went to Salt Lake City and felt like garbage for a couple of days. But then I kind of bounced back. So I'm not sure about the relationship between that and this, but what this tells us is that bisphosphoglycerate plays a role in basically helping you in a low oxygen environment. So the atmospheric pressure decreases with altitude, which is what we see when we go up to 4,500 meters. And in the short term, your body's going to make more BPG. And essentially, this is all of what I said. Okay, now here we go with a question. A newly discovered compound causes hemoglobin's affinity for O2 to decrease. Which of the following is true? I want you to think about this in terms of all of the different terms that we've used and the different ways that we've described hemoglobin and characteristics and properties of hemoglobin. So we've got 52 seconds. Ten seconds. I'll bump it for another 15. So we had about 36 people answering. So 15 seconds. 2 seconds. 1 second. Okay. So when you look at this question, there's a couple of different things that are kind of presented. And this is why I think that it's very important for you to really understand and to study an oxygen binding curve. An oxygen binding curve, our y-axis is that y or theta if we're talking about any ligand, but we're talking about oxygen binding proteins. You have 1.0, 0.5 as our big landmarks or our benchmarks, and then along your x-axis is your PO2. And that PO2 is basically the concentration of oxygen in the environment. That's something that you don't control, or that you control, and the protein behaves in response to that. Now, if we were to see a myoglobin binding curve, it's going to look a little bit like that. I apologize for my drawing there, but what I need you to be able to pick out is the P50. The P50, and I always like to bring this up, is that the P50 has unit values, and it has unit values of torr, the oxygen concentration, or the oxygen, the partial pressure of oxygen. So 5 torr, for example. So this myoglobin protein that I've displayed here has a P50 of 5. Now this curve can shift to the left or it can shift to the right. Before I continue, let's go ahead and check the correct responses. Okay, so we got a good peppering of answer options. E is the correct answer. The T state of our protein is favored. So hemoglobin's, we'll dissect the question. Hemoglobin's affinity for O2 decreases. Okay, so that means hemoglobin's kind of request to bind O2 goes down, which means that we're favoring, favor the deoxy state of hemoglobin, or deoxy-HB. Now, that deoxyhemoglobin is also known as the T state. Now, those two terms are kind of interchangeable. The reason that we have those interchangeable terms is because Hemoglobin is an example of an allosteric protein. Allosteric proteins, all of them, have T states and R states. The T state is your inactive. In this case, it's the deoxyhemoglobin. The R state is your active form. So with this compound, we're going to favor deoxyhemoglobin. And so that is going to dismiss answer option C. We're going to favor the T state by virtue of favoring the deoxy form. Okay, so there's that. But then what about these two answers here? The P50 of hemoglobin decreases and the oxygen binding curve shifts to the left. I want you to be able to see this question or see these answer options and understand that they mean the same thing. They look like they mean something different, but they actually mean the same thing. Okay, so just to illustrate that, let's say that I drew a curve, effectively the same shape, but it shifted to the left. What would that look like? Okay, so here's our P50. Our P50 is now, let's call that 1. So when the curve shifts to the left, you have to look at that coordinate. One thing that I've run into in the past that people get, and whenever I say people, many times I'm meaning younger Joel Gray. What younger Joel Gray got confused about with respect to P50 was if you asked me what the units were for P50, I would say, like, I remember the first time I was asked that sort of question, I said, percent. Not really. Percentage isn't a unit. It's a reflection of a ratio. What we need to do is coordinate that percentage, that y-axis, to our x-axis, which is our unit, our measure, or our unit that we're measuring, which would be, in this case, torricelli, so the amount of our gas. So it's important to, whenever you see this sort of question, be able to critically look at answer option A, and then look at B, and see that, oh, they say the same thing. One of them, you see the curve shift, but what does that shift of that curve mean? Okay. Now, the next thing, and I gotta just a second check this. Okay, cool. That's where I wanted to be. Okay, so fetal hemoglobin is the form of hemoglobin that a fetus has. A fetus receives O2 from its mother's blood through exchange or through the placenta. Now, fetal blood has a higher affinity for O2 than the mother's. So you might think, oh, well, they're going to be fighting it out. And well, fetal hemoglobin is different structurally from adult hemoglobin. Adult hemoglobin, remember, has that alpha 2 beta 2, so it's a dimer of dimers. Well, fetal hemoglobin is as well, but the difference is rather than it being 2 alpha and 2 beta subunits, fetal hemoglobin has 2 alpha and 2 gamma subunits. So the distinction there between the gamma and the beta subunits is a couple of amino acids and a little bit of their structure that makes them different. Now, the intrinsic oxygen affinity for HbF, or fetal hemoglobin, is very similar to HbA. HbF, however, has a much lower affinity for bisphosphoglycerate. So any amount of bisphosphoglycerate that's produced by the mother is going to impact the amount of O2 that it binds. So then all of a sudden, whenever bisphosphoglycerate is present, then the fetus or fetal hemoglobin is able to bind that O2 because of the presence of that bisphosphoglycerate. Now, the distinction on a molecular level is that hemoglobin or the adult hemoglobin has a histidine at position 143 in the beta chain, in the beta subunit, and fetal hemoglobin has a serine at position 143. Now, if you think about those two amino acids, we've got a polar positive residue and a polar uncharged residue. Now, the change of a positively charged histidine for a neutral serine reduces the binding affinity for bisphosphoglycerate to HbF, because remember, bisphosphoglycerate mediates its interactions with hemoglobin through charge-charge interactions. Now, I did have a last question posed, but I'm going to go ahead and save that. We're going to kick class off on Friday with that first question, or with that last question from today. But before I open the floor for any questions, I wanted to say that I've been working on a practice exam. So I've been writing your exam number 3 and this practice exam alongside one another so that I make sure that one, I'm following the learning objectives that I provided for you, but also that in this practice exam, I'm giving you questions that reflect what are going to be on the actual exam. Now, I'm going to be publishing that practice exam hopefully before the end of today. But what I want you to know about that practice exam is that whenever you see the questions on there, or whenever you're going through the questions, what I want you to think about is how could I take this question and make it slightly different? And I know that that's how I've posed or proposed that you look at questions in the past. But if this question asks about the alpha subunit, then say, okay, what if I change this question to the beta subunit? And that's just a hypothetical example. But right now, what I'd like to do is I'd like to go ahead and stop my recording.